Welcome everyone. Thanks for the interest to this talk. I am Jamal Koba. I'm a fairly new postdoc in Sano, and there are also already some people that I met, but some of you I don't know. Nice to meet you. And today I will talk about effects of stroke on the brain connectivity gradients. This is my project that is in process. I think to start with, since your attention is already fresh and you haven't started digesting the pizza, I will start with the functional connectivity gradients. Functional connectivity gradients is a method to measure the connectivity profile of the brain. And unlike the traditional functional connectivity networks methods, like seed-based connectivity or ICA-based connectivity, this method is useful to show the structural and functional organization at one dimension. And this is inspired by, I'm quoting the authors, this is inspired by the gradients of structural measures such as cortical thickness, myelination, and cell microarchitecture in the brain. And on the left side, you can see the similarity profile of the cell architecture. And on the right side, you can see the similarity profile of the myelin ratio of the cells of the brain. And in both, you can see a distribution of transmodal to unimodal regions, meaning that unimodal regions that are mostly associated to a specific cognitive tasks such as vision, motor functions, or hearing are closer in the connectivity space compared to the regions that are related to the more cognitive functions, let's say. And authors say that they were thinking about this distribution, and they wanted to see if they can create a map for the functional hierarchy of the brain rather than the structural hierarchy. And they did it, but they had a very big challenge actually, because when you map a structural measure, there is one measure for every cell or every voxel or every point in your measurement. But as you know, functional part of the brain, functional fMRI, has more than one time point for each region. So there is like a more than one dimension for each point. So in order to make one uniform map, they needed to reduce the dimension of their data. And they did it via a nonlinear dimensionality reduction technique called diffusion embedding, which I will come to it later. And they managed to relay the map of functional macroarchitecture of the brain to the whole cortex. And not surprisingly, this map also is in line with the other structural measures such as cortical thickness, number of neurons per region, or even gene expression. So they concluded that the most variant or functional hierarchy in the brain, or at least most of it, can be explained by the structural background of the brain. So it is not completely different or random. It depends on different parameters such as cell type, number of neurons per region, or like gene expression. And how do they do it? How do they do it is, up until some extent, it is very close, actually identical to the other traditional functional connectivity calculations. First, they take the fMRI data from the whole brain, and they extract the time series. They clean the data, they extract the time series, preferably voxel-based, because if you are going to make a gradient, you will need a high resolution rather than like big chunks of areas. And after getting the time series, they created the functional connectivity matrix you can see on the bottom left. And up until here, it is more or less common for all connectivity studies. But then what they did is they calculated the cosine similarity of the regions based on their connectivity profile. So they checked how similar two regions in terms of connectivity, if their connectivity profile are with the other voxels are similar or not. And then they this resulted in the affinity matrix, which is actually also a very basic transformation technique. Most of you probably know better than me. But still, there are like too many points to relay it on the map. So they needed to reduce the dimension to the number of voxels or the number of vertices on their model, right? And they fed the affinity matrix into the diffusion embedding algorithm, which puts the data to a 2D or 3D dimensional space based on their values of affinity matrix. So this is the result of the diffusion embedding process. And as you can see, the regions that are that have similar values on the first axis, which explains the most variance in this matrix, are here. And when you rely on them on the brain, these are those regions. So you have to see both ends of this axis, and you can see on one end there are the red regions. On one end there are the green regions. So this is what the algorithm gives you. It doesn't tell you how they are different, or what is there on the both ends of the axis. It just tells you they are different in this way. So you have to figure out what is there. And when you look at it, it is still the unimodal transmodal distribution. Some also call regions that are task positive, task negative, or like the default mode network or task positive networks, which is in line with the other distributions that we mentioned at the beginning. And then you can also check the second gradient that explains the most variance, and you will find what they found was distribution between visual regions and sensory motor regions. So they concluded that brain functionality mostly is categorized in task negative, or visual sensory motor regions. And on the rightmost side, you can see the sorted connectivity matrix based on the affinity matrix. Is everything clear? I mean, I'm trying to make it as clear as I can, but I am also just getting into the literature. So if anything is missing, just let me know. So this was the functional connectivity gradients. Oh, some more details. Yeah, it's just like another way to look at the data. When you relay, when you put the connectivity gradient on the functional parcellation, you can see that the regions. The first gradient is on the axis of task positive and task negative. Second gradient is between visual and somatomotor. So brain functionality divides itself in this way, but based on what the researcher has to find out. Okay, then here stroke comes in. Why stroke general description? It's a phenomena that occurs when something either blocks the blood supply or blood vessel in the brain bursts. So either there is lack of blood supply or too much blood supply that destroys the vein. And of course, there are different reasons for these, the risk factors and everything. But what I am interested in is why it can be a good model for studying functional connectivity. So actually, the answer is pretty easy. The lesion studies had always been important since the beginning. Even Wernicke and Broca area were discovered in this way. So a big data set of stroke or other type of lesion can be like a big opportunity to study the disconnectome of the brain rather than the connectome, which is a term that is already being used in the last years. Researchers are trying to map what kind of regions lose their functionality when there is a problem in a certain brain region. And in some sense, the cases like sensory deprivation, for example, think about ocular blindness. You can study how the plasticity, apart from the physical disconnectome, you can study how the plasticity changes in response to lack of modality or visual input or like hardwire problem. You can also see the plasticity in response of that problem. But this stroke is more unique because every case is a case study. Every case is a different story. So their lesion volume is different, effect of area is different. So actually, if it's like a treasure waiting to be discovered. And of course, MRI is a great way to measure it, which brings us back to the functional fMRI and functional connectivity gradients. And of course, stroke had been used to study brain connectivity, functional connectivity in more traditional sense, the importance of the location of the lesion, and the plasticity, or like how, if brain tries to recover itself, or if the disconnection between functional disconnection between regions become worse over the weeks or time, has been studied, but not from the perspective of the gradients. And yeah, there is only one study actually that both inspired me and made me want to add something on it if I can. They checked the effect of stroke on the connectivity gradients in a very straightforward, basic way. I will just talk about their main methods, and their main results. Of course, there are more different tests that they applied, but all based on this methodology, so just a byproduct of this methodology. And what they did was, they have two groups, stroke lesion group and healthy control group. And first, they calculated the functional connectivity gradients for the healthy group. And they pictured the first three gradients that explained the most variance in the functional space. And they managed to replicate the results of the first authors, or like general results that I mentioned at the beginning. But what did they do with the stroke patients was not very direct. So instead of comparing the gradients from stroke and healthy, they just took the lesion masks for each stroke patient, and they calculated the mean diffusion embedding in that mask on healthy data, on the healthy template. And for the rest of the brain, for each voxel, they subtracted the real value from the mean value in the lesion mask, giving a distance to lesion map. Remember that they are doing this calculation on the diffusion embedding score on this. So the difference actually gives the connectivity profile difference between the lesion region and the other voxel, non-lesion voxel. And on top, they created this for each subject as a complementary analysis. They had the longitudinal data for stroke patients, scanned after the first day of the incident, stroke, second day, and fifth day. They calculated the same for each day, but for each voxels, they calculated the concordance value to see how the connectivity profile changed over time in time span of 5 days. Concordance value is kind of a correlation that shows how similar are the scores. If the value is 1, it means that the values are concordant, they do not change over time. If -1, they change completely. The problem here is that it doesn't say in which direction it changed, like positive, negative, if the network is disrupted, or if network is just repairing itself. And then, after they had this concordance map showing the consistency over time for the functional connectivity profile, and distance to lesion map that is calculated on the healthy subjects, for the whole brain, for every voxel, they calculated the correlation, basically, between concordance and distance. And this created one value for each subject. And this tells how, it's a bit like tricky to conclude, and this tells if the connectivity profile of a voxel is different from the lesion voxel, and if that profile is stable over time. But there are some methodological problems in my opinion, because they were both assessed in different groups. Okay, as a result, what they found was on the right side, for the first gradient, they found a significant mean correlation of 0.6, if I remember correctly, and significant correlation, positive correlation in gradient 3, and no correlation in gradient 2. And they concluded that since the first and the third gradients separate the functional organization in task positive, more like task positive, task negative, or different networks. It was expected to have significant outcome, but since the second gradient separates the brain between visual and sensory motor cortices that are like not functional connectivity networks, but they are more like distinction between unimodal areas. Of course, they will not reflect the features of the functional connectivity networks in covering the whole brain. So this was their conclusion, and they drew some like messages from these conclusion, but I think all you can say from this result and with this data is for healthy subjects, the connectivity profile to the lesion mask taken from a stroke patient is different, and for the stroke patient, that connectivity profile changes over time. So it is very indirect way to measure it, and I think making some conclusion is very hard. So since I also started working this area, this paper inspired me. They also said that it is, the authors also said that it's like a beginning primitive way to start checking the gradients in stroke, because it's tricky, because like lesion masks are everywhere and it's hard to get a whole brain connectivity pattern. And from this work and from the previous other works that are related to connectivity analysis with stroke, there were some points that attracted my attention and would like to take them into consideration for my work, which is first of all, identifying the lesion in form of mask only is not enough, because it's been clear that there are like distant regions that can be affected in a single point of stroke or like a damage, and you have to take this effect on the other or distant regions into account. Otherwise, you will not have a clear calculation of the gradient. For example, on the leftmost side, you can see it's already a toolbox. It's a like generally accepted method or study. If you give the lesion mask on the standard space, it will tell you the possibly affected white matter tracts for the rest of the brain. So actually, ideally, what you will do is, if you want to block the lesion mask, you want to block that one too, right? Ideally, but I will discuss it later. Also, there is another study calculated the affected both cortical and white matter parts by themselves. So it's still a stroke study, and they take into consideration this possibility. And there is another study and toolbox on functional and structural connectivity of a given voxel. For example, if you give a voxel in the visual network, it will give you the functionally cortically relevant gray matter areas plus white matter structures relevant to that functional connectivity network. So I think before beginning, this information should be taken care of and saved somewhere, because in terms of censoring the lesion mask, this should follow and this should be put in the model too. because yep, we are with functional connectivity gradients, we are reducing the dimensionality a lot, and a lot of information is lost meanwhile. These must be in the model to account for these problems. Another thing that I would like to check is temporal features of the bolt signal, because stroke, most common type of stroke is the ischemia, and ischemia is the lack of blood flow, or blood clot in a vein, which means that the fMRI won't be able to extract proper bolt signal from that region plus the relevant regions. And according to the literature, they say it is okay with the resting state fMRI, since you just compare one group with another. I think that not between group, but within brain, it can be a problem, because some regions will give regular signal, and the regions that are not affected by the stroke will give a different kind of signal with different temporal characteristics, which can disrupt the correlation over the regions. And this example is from a task-related fMRI, task-based fMRI. On the areas that have gray shade, there is the task. And you can see on the left side for the controls, it is very nice and clear. But on the stroke patients, from the stroke region, it's quite disrupted. I think it will be also seen in resting state fMRI, although I haven't tried and I couldn't find anything concrete on this matter. But it should be taken care of. Also, because there is something called arterial labeling, and this arterial labeling shows the map of what kind of arteries feed what part of brain. So let's say that a blood clot occurs somewhere here, and then that artery keeps branching to the rest of the area. Apart from the functional or white matter connectivity, because of the arterial connectivity connections, the rest of the region will have disrupted bolt signal either. And luckily now they have this labeling system on NIfTY format, neuroimaging format too. So I'm actually planning to take this also into consideration as a third type of connectivity, rather than in addition to white matter and functional connectivity information. Also, in the study I presented, it was the first thing that I saw was they didn't calculate the connectivity gradients for stroke patients. Rather, they used an indirect way to calculate. But I think they must be calculated. And I think the author's hesitation was the difficulty of creating a whole brain gradient in case of lesion masks on the cerebral cortex, gray matter area. But this is the very accurate representation of the mean stroke region. So strokes are not actually randomly distributed over the brain. They are mostly around the thalamus or basal ganglia region. So actually, most of the stroke regions are in subcortical area. So it is still possible to create a whole brain connectivity gradient and compare them with the healthy. But since all lesions are not exactly the same and different, it wouldn't still give an accurate group difference. So individual region of the lesions must be still accounted for. But that would, if we used only the subcortical lesions, that would only give us a title such as the effect of subcortical stroke on the cortex. But we also want to make use of the problems on the gray matter, which I will come to it soon. And in the study, again, the longitudinal effect was not clear. The change on the connectivity gradients over time was not clearly defined if it was like positive or negative, or disruption or recovery. So I think I will also take this into account, like the direction of the change. This is all from me. Thank you for listening. If you have any corrections, suggestions for my possible work or questions, I would love to hear.